IQ Microphysiological Systems workshop series. Today we will be hosting several companies presenting about blood-brain barrier microphysiological systems. We are always excited to have the chance to create these connections between our tech developers and other stakeholders across industry, academia, and government. Now, before I get started, I do need to read this disclaimer. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in the webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including things like cost, prices, sales, product marketing, and other confidential information. In addition, neither the 3Rs Collaborative nor IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. Now, for those of you joining us for the first time or watching this presentation as a recording after the fact, I wanted to give a brief introduction to the 3Rs Collaborative. Now, our group is a really special nonprofit dedicated to improving animal-based research through the 3Rs: reduction, refinement, and replacement. And our mission really is to advance better science for both people and the animals involved in animal research. Our Microphysiological Systems Initiative group is a big part of this, and in their efforts, we are hoping to increase the adoption of and regulatory acceptance of microphysiological systems technology in coordination with animal studies. Our MPS systems group is primarily comprised of technology developers, but we are proud to have a diverse array of individuals from academia and governmental bodies additionally. Now, the 3Rs Collaborative Microphysiological Systems Initiative aims primarily to do 5 things. First, to provide thought leadership and expert consensus from our tech developers and other stakeholders on the implementation of MPS technology. We aim to facilitate discussion, collaboration, sharing, really connection between our commercial developers and other stakeholders. We try to create direct engagement between the developers and regulatory agencies on the use and uptake of microphysiological systems. We aim to develop external partnerships and collaborations across the field to improve practices for MPS developers. And then we aim to create resources to facilitate and educate and increase engagement between our tech developers and other stakeholders. To do this, we have 4 primary work streams. First, we interface extensively with end users by coordinating webinars, workshops, and other activities. We connect our developers and regulatory bodies by primarily by increasing awareness of the issues surrounding MPS acceptance. We have created and maintain a technology hub on our website that provides the ability for our tech developers to present their technologies to potential end users and for those end users to search for the technologies that are most appropriate. And then we aim to create extensive education materials by presenting at conferences, creating posts and other digestible resources for end users to better understand how MPS technology could be useful to them. Of course, we are constantly engaging with end users, and over the last couple of years, we have done this through our webinar and workshop series, of course, but also through presentations and connections at major conferences around North America and internationally. Our MPS group has a specific subgroup focused on engaging with the regulatory side of MPS technologies, and they have been very busy over the last couple of years promoting and connecting tech developers and regulatory bodies. And then, of course, we are educating end users and other stakeholders about the value that MPS can bring to a research project. And all of these are things that we are planning to continue this year and continue to expand. So we are really excited to kick off our workshop series with our blood-brain barrier companies. If you are interested in more information from our tech developers, you can visit our technology hub at the North American 3Rs Collaborative website. And here you can use filters to drill down on various things that will direct you to companies with the products that you are needing. Of course, this isn't our first workshop, and you can view the previous presentations on our website. I'd encourage you to go back and look at those. They cover a number of other tissue systems and microphysiological technologies that I think could be of significant interest. If anyone has questions, please put them in the Q&A section of our talk today. You can find that at the top of the blue arrow. But if you put it in the chat, I will still find it. At the end of all of our presentations, I will be facilitating a roundtable discussion with our companies to answer any questions that we might have about blood-brain barrier MPS tech. And with that, I will happily turn it over to my colleague, David, who will introduce you to the International Consortium for Innovation and Quality in Pharmaceutical Development. We are really happy to partner with them in this workshop series. And with that, I will turn it over to you, David.
Very quick. Okay. And yeah, I'll try to go quickly so we can get into these great talks. So hi, everyone. My name is David Kukla, and I am part of the IQMPS affiliate. And I specifically serve on our strategic partnerships and communications team. So yeah, I just wanted to give a quick overview of the IQMPS. So the IQMPS is currently composed of 22 member companies that you could kind of see over on the right in the teal. And we have representatives from drug safety, 3Rs, ADME, and PDPK. And our leadership team is made up of Rhiannon Hardwick and Anya Kopek. And soon I'll be able to update everyone on our 2023 leadership team. But besides that, we have a steering committee that is made up of 2 individuals from each of the 22 member companies. And there are 4 main goals of the IQMPS. One being to serve as a thought leader for both MPS developers and stakeholder organizations, provide a venue for appropriate cross-pharma collaboration and data sharing, to create a focused engagement between industry and regulatory agencies, and develop external partnerships and collaborations to help enhance the inclusion of industry priorities. And here are the IQ MPS work streams, including manuscript publication, publications, regulatory outreach, proof of concept studies through pilot project teams, strategic partnerships and communications team, as well as a landscape gap survey team. And here's just an overview of our external partnerships over the past few years. And for the sake of time, I'm not going to go into all of them. But within 2021, we co-partnered with the NA3RC and established an MOU, which allowed us to work together on webinars and workshops, such as this workshop today. And we're really excited to continue this partnership into our third year. Besides that, we've engaged with several different external contacts, and we've provided a year-end update and overview landscape assessment through surveys. But yeah, that's all I kind of have for right now. But feel free to reach out to me if you have any follow-up questions on the IQ-MPS. But yeah, we're really excited to get into some great talks. Thank you.
Thank you so much, David. And yeah, my apologies for going over, but now we're really excited to introduce Chris Hughes from Aracari Bio, and he will share with us some of his company's technology. So Chris, I will let you introduce yourself a little bit more and go ahead and take it away.
Thank you. All right. Morning, everyone. Just getting used to Teams again. All right. Can I get some confirmation that you are seeing my screen?
Chris, yep, we are seeing your screen just fine.
Great, thanks very much. Morning, everyone. I'm Chris Hughes, Chief Scientific Officer at Aracari Biosciences. I continue to hold a position as a professor at UC Irvine as well. And some of the work that you'll see is being developed in the academic environment, some of it inside Aracari. So this is what we're talking about today, is the vasculature of the brain. There's a lot of it. It's highly complex, and its major role is to protect the brain from fluctuations in nutrients, toxins, whatever, in the blood. And this is the classic experiment that I think we're all familiar with, which is the injecting Evans blue into a mouse, then taking a look inside that mouse. Most of the mouse goes blue, except for the brain and a couple of other places that stay white, and that's because Evans blue, which binds to albumin, doesn't freely cross the blood-brain barrier, demonstrating protection of the brain. But if the brain is traumatized or damaged in any way, then the blood-brain barrier collapses, and then you get free perfusion of the brain tissue with whatever's in the blood, and that's shown on the right side of this image. So a sort of a graphic representation of what the blood-brain barrier is all about. So some of you may or may not be familiar with the basic Aracari technology, so I'm just going to give a quick summary of that. What we, and I think most of us, are trying to do is to hit this sweet spot between simple systems and physiologic models. At one end, obviously, we have the very definition of physiologic, which is the human. And at the other, we have what we're all familiar with, which is 2D monolayer monocultures, which are simple to use, but in many cases are not physiologic because the lack of the third dimension and the lack of additional cells to interact with, and in the case of the vasculature, a lack of physiologic flow. Other model systems around, we're all very familiar with the mouse. Many of us are here for the explicit reason that we're uncomfortable using animals and want to see alternative and in many cases improved models, because mice are not people, and much of what we've learned from mice is not as translatable as we would like it to be. And we also have spheroids, which can be quite useful in some instances, but fall down in others. Our technology, as I said, sits in the sweet spot. We generate vascularized microorgans. What you're seeing here in red is living human vasculature. think everyone's familiar with the microfluidic channels representing the artery and the vein, top and bottom, and then this capillary network that connects the two. And within this space, we can grow any tissue we like. And as you'll see, in this case, we'll be putting cells derived from brain. So again, vasculature, artery, vein, capillaries. This is how we schematize it, again with the microfluidic artery, a microfluidic vein in blue. We can drive flow through this, as we'll see, and we connect these with, as I said, a living human capillary network. All of this sits in this diamond-shaped tissue chamber, and within here we put initially a slurry of cells and matrix, and that matrix is tuned to whatever tissue we're interested in. So in this case, it'll be tuned to the matrix of the brain, so that's laminin, hyaluronic acid rich. The platform has been miniaturized, so we can put these tissues on a 96-well plate. You can see these line up nicely with the wells. So here's our diamond-shaped tissue chambers lining up with one of the wells of this 96-well plate. We currently put 16 individual tissues on one plate. Each of those are individually addressable tissues. And you can see down at the bottom there how tissues grow nicely in this environment. And this is the vasculature. You can see that it's tubes forming here that connect top and bottom. The cells are transduced with GFP, so you can see them. In many cases, they have M-cherry. So why do we go to all the effort to vascularize organs? We could just make a monolayer blood-brain barrier. All organs are vascularized. Vessels actually help to pattern the tissues. So there is crosstalk between the vasculature and the cells that surround it, independent of what the vessels are carrying. So the endothelial cells themselves crosstalk with cells and tissues. Most cells, as we know, are close to blood vessels, and all nutrients and drugs are delivered through the vasculature. And also, as I mentioned, there's flow and there's and shear actually has quite a dramatic effect on gene expression in the endothelium. So to best model human disease, we really need vascularized and perfused tissues. So just going to show you what the platform looks like. So here's an artery, the larger vessel, and you can see the capillaries springing off that. We can run blood through this. This is blood in the skin, and you can see there are areas of low flow and areas of high flow. The flow rate can be tuned. We use hydrostatic heads, not pumps, so we can dial in any flow rate we want just by changing the amount of medium in the driver wells. Importantly, parasites do wrap around the vessels. This wrapping is flow dependent. Under flow conditions and appropriate shear, the endothelial cells will make PDGF-B. PDGF will then recruit parasites to the vasculature. The base platform that we use shows physiologic permeability. So here, this is 70 kilodalton fitsidextran, and you can see that that slowly leaks out over time to fill the chamber. If we put in 150 kilodalton fitsidextran, so this is around the size of an antibody, you can see that that comes out much more slowly, and we can calculate the permeability coefficients, and those match what you'd see, for example, in skin capillaries. And we can challenge these vessels. This happens to be LPS, and we get a dose-responsive increase in vascular permeability. So in all ways that we can tell, these vessels are behaving in a physiologic manner. So in terms of blood-brain barrier, we have a version that we currently offer and a new version that is coming along. I'll talk briefly about both. This is the structure that we are trying to recreate. The neurovascular unit, the endothelial cells in green, connected by tight junctions. We have a complex basement membrane around that. Embedded in and around that are perivascular cells such as pericytes, and then astrocytes, as we know, put down foot processes onto the surface of this structure to help maintain the blood-brain barrier phenotype. Currently, we, like everybody else, uses all-trans retinoic acid to induce a blood-brain barrier phenotype in endothelial cells. We use cord blood-derived endothelial cells, and those, as you'll see, will differentiate quite nicely. So in these images, you can see the endothelial cells. The top two images are in green, and you can see astrocytes in red with their typical star-like pattern, and you can see those putting down foot processes onto the abluminal surface of the vasculature. The bottom left, the endothelial cells in red, you can see pericytes in green, nicely wrapping around those. And again, astrocytes putting down foot processes. The bottom right, this is a stain. GFAP is a marker for astrocytes in green, and you can see a collagen 4 rich basement membrane there. We can also incorporate neurons into this. These are human neural stem precursor cells in green. And if we play around with the medium a little bit, we can find that we can get all the cell types behaving appropriately in the platform. The vessels do polarize, as you'd expect. So we have a collagen 4 rich basement membrane in white, surface expression of ICAM1 here. I'm not going to show the data, but we can get very nice leukocyte extravasation across our vessels. And then on the right, again, you can see pericyte wrapping. Pericytes are a critical component of the neurovascular unit, and it's important to see that these do wrap appropriately around the vessels. This tight apposition between the endothelial cells and the pericytes is crucial for proper blood-brain barrier function. We've also looked at tight junction expression. Here's ZO1, Claudin 5. Those junctions are present. And again, the basement membrane is important. This is collagen 4. We've also looked at laminins, which are known to be a component of the basement membrane in brain. And here's staining for alpha-1, alpha-2, and alpha-5 laminins. So gene expression, this is some qPCR we've done. Again, the genes of the sort of things we're interested in, GLUT1, P-glycoprotein, other drug transporters, tight junctions. These are upregulated in our neurovascular unit. Some key genes that we've looked at here. Everyone can pick out their favorite blood-brain barrier genes, but as you can see, many of those are expressed here, as you would hope. And critically, the permeability is dramatically reduced under these conditions. That's our regular vessels that I showed you before with the white squares, and that slow leak over time. And our blood-brain barrier vessels show a dramatically decreased permeability to fitsidextran in this case. So we've performed single-cell RNA-seq on this platform to further understand it. Again, this is the neurovascular unit that we're talking about. The image there, this is the kind of tissue that we isolate. So you can see we have all of the cell types in there. We use 10x genomics chromium system for this. And then we compare gene expression to a human ventral midbrain data set that we pulled down off the web. So the smaller plots here are t-SNE plots for both the human ventral midbrain, top right, our VMO, blood-brain barrier, or VMB as we sometimes call it. There, the integrated data set is on the left. And based on gene expression, we're able to identify this green population circled as the endothelial cells. And then on the right, the gene expression in the brain tissue in this orangey pink color, and our platform in white. And you can see nice enrichment in both of transporters such as SLC2A1, drug efflux proteins, tight junctions, MFSD2A, which we know is involved in suppression of pinocytosis, also nicely upregulated here. So in all these ways, it looks like we generated a nice, fully functional blood-brain barrier. That said, we are not super happy with the use of all-trans retinoic acid. It takes quite a large concentration. In our hands, and I think in most people's hands, the effect is transient. If you take the ATRA away, over the next few days, the barrier properties will decrease back to baseline. We're looking for something that lasts longer, and so we're developing a different version of this. Again, the same structure here that we're trying to generate. This is our version 2. These are physiologically induced cord blood endothelial cells, again, not using ATRA. We can put in neurons for staining there for beta tubulin. That looks very nice as well. We get, again, astrocytes attaching to the basement membrane. We get parasite wrapping. We get a very tight barrier. As you can see, there's almost no leak to 10 kilodalton fitzy-dextran over 30 minutes. And most importantly, this phenotype is sustained over multiple days. We now have a new design that allows us to access the CSF compartment. So we can look at transport across the barrier into the CSF. And that's just showing here. We've also got a new plastic. We've moved away from PDMS. This is just showing that if you put citalopram in one end and measure the output at the other end, none gets out in PDMS because it gets absorbed, whereas our new plastic in gray, most of that does make it through to the other side, which is important to know. And we're also
Dr. Hughes, I'm sorry to interrupt, but we are running out of time. Okay, thank you so much.
This is my second to last slide. So this is establishment of gliomas. We're also interested in how that affects blood-brain barrier. These are the kind of readouts that we can do. And just to summarize, these are fully human tissues, physiologic permeability, fully functional blood-brain barrier. Thanks very much.
Thank you so much, Dr. Hughes. That was really, really interesting. And next up will be Wouter Strijker. Wouter, if you can join and share your screen, I will let you take the stage.
Cool, thanks so much. And indeed, another check if you can see the screen and if you can hear me.
Looks great.
Perfect. There we go. So I'm Wouter, I'm part of the business development team at Mimetas. And today I'll briefly update you on the BBB models in the organoplate. And just as a brief introduction on our company, I have one slide on that, and the rest I promise is all on BBB related data. But just as an overview, so we are in the game, and I'm very glad that many of us are in the same game. Is a nice screenable platform that we work with. We developed unique assay capabilities with translational readouts that I will show a couple of throughout this presentation. We've worked with many of the top 50 pharma companies in the world and really provided proven impact on their decision making. And we have a team of around 120 people right now, most of us in Leiden in the Netherlands, but also with facilities in the US, in Maryland, and in Tokyo. And today we will be talking about blood-brain barrier. And actually, you can see that in many different ways. I will start with some examples on our monoculture model that we develop in the organoplate tree lane. And later on, I will also show a couple of co-culture systems. So looking at the monoculture, there we work with an organoplate, as mentioned, where we have 40 or even 64 chips on one plate, each consisting of 3 lanes, where we use the middle lane to seed an ECM gel, and use one of the outer channels to seed endothelial cells that upon flow form very nice vessels, which can then be perfused with medium. And it's good to realize that we also have a third channel, which we keep empty, or we add medium there as well, which can nicely be used as a sampling way, providing access to both the apical side of the tubule as well as on the basolateral side. We are working with primary human brain microvascular endothelial cells. It actually took us quite a while to get to that source. After first trying different cell lines, different IPS-derived endothelial cells as well. But neither of those was very successful in our hands. The cell lines, we couldn't get it as leak tight as we were hoping for. And for the IPS-derived cells, we actually saw over and over again that they formed very nice leak tight barriers, but we couldn't maintain the endothelial phenotype. So over time, the IPS cells always showed epithelial-like phenotype, but not for these primary brain endothelial cells that we optimized. You can nicely see by these marker expressions that endothelial markers were expressed, like PCAM, via-cadherin, as well as tight junction markers. We could form very nice barriers. So here an example of a fluorescent dye that we added in the lumen of the tube. These two are examples of no cells, and in the others you can see that it nicely maintains within the vessel. And we could show relevant BBB transporters like PGP, but also MRP1 and transferrin, for example. And for us, we see use of this model in 3 main applications. One of them is barrier disruption for toxicology type of questions. Another one is looking at drug transport, and a third one on disease modeling. Now I will show a couple of examples of each of those applications today. Starting with barrier disruption. So we can nicely track the barrier of the endothelial vessel by measuring TER, so transendothelial electrical resistance. We do that with our organotear device shown on the right bottom of the slide, where you basically place an organoplate in the organotear, as we call it, connected to a computer, and the software would allow you to measure the TER values of the 40 or 64 chips all at the same time, and track that over time as well. So here, what we've done in this study, we looked at a couple of compounds that were actually suggested by one of our pharma partners, and looked at the TER values over time, and see what kind of effects they would have on the disruption of the barrier. So if we start at the top, so TNF-alpha and LPS, for example, nicely showed dose-dependent TER drop over a time of around 18 hours. Also for NMDA, you could see a nice drop, not really dose-dependent to be honest, but you could see a nice drop in TER. And the 2 also very interesting ones there are at the bottom, where thrombin is known to have a transient effect, and especially in the high concentration, you could see a nice drop in TER in the first hour after exposure, but it's recovering over time, not up to the level of the medium control, but you do see some recovery over time, and that's also a very known effect of thrombin. And then the VEGF exposure also showed a clear effect on TER values, and that was a very good affirmation that we were working with proper endothelial cells, since it's vascular endothelial growth factor VEGF. So it was a good confirmation that we are actually working with endothelial cells. And this is now by that pharma partner that I was mentioning, who suggested these compounds. That company is now using this for routine use as well. Then looking at drug transport, and there the importance obviously is for, especially if you're looking at small molecule transport, is the tightness of the barriers. So here we have a control of a full plate with 2 cell-free controls, and the rest all with the barrier as mentioned. And we added a small molecule sodium fluorescein, and you could see that that basically this plate is QC checked, because all the chips with cells in it could be used. They are strong enough, the barrier is tight enough to maintain the small molecule within the vessel. And on top right, another representation of such a chip. I think it's nice to realize that we now have an option to add small molecules to the lumen of the tube and then sample on the basolateral side of the tube. And there is no interference of artificial membranes or whatsoever. The only thing that the small molecule need to pass is the actual barrier of cells and the ECM compartment. So this is what we've done with another pharma partner actually. They provided us with 18 compounds, of which they knew whether it was low, moderate, or high BBB permeable. We added that to the lumen, took samples on the basolateral side, and analyzed that by mass spec. And although we switched 2 compounds around, we could rank this as expected based on in vivo as well as clinical data available. So we were very happy with this result. And then the next step is to look at antibody transcytosis. I think this is something super relevant for many companies right now developing antibodies against CNS diseases. Is to what extent gets the antibody to the brain compartment, obviously. So it's the same approach, adding it to the lumen of the vessel, and then taking samples on the basolateral side, and doing MSD or ELISAs to then measure the antibody transcytose. And here, this is an experiment of two antibodies, where we actually first look at the amount of antibody that we could see on the basolateral side, on the left side of this graph without cells, and on the right side with cells. So I think it's interesting that you can already see that these cells have a huge effect, obviously, on maintaining the antibody on the apical side of the tubule. And you can also already see a difference between positive and negative control. The only thing that was also striking to us is that there seems to be some differences between these 2 controls that we selected on their permeability throughout the ECM. So if we correct for that, then you see indeed that the ratio between positive and negative control in the cell-free is not exactly 1. So if we correct for that again, you get a ratio between positive and negative control antibody of around 2. And this was targeting the transferrin receptor. And I think also in literature, this is the twofold difference is also as expected. And now obviously we are also targeting different transporters to see if we can get an even higher fold difference between antibodies. And then the last application that I would like to focus on is disease modeling. And there it's obviously very interesting to not only have your endothelial cells present, but also a brain compartment, so to say. So here we have a system with endothelial cells, again in the same way, perfusable, and then in the 3rd channel we added neurons and astrocytes to create a triculture of those cells. And then we started to trigger that in different ways to basically model a stroke on a chip. And we combined that with low glucose, chemical hypoxia, and low flow. And the different triggers all had an effect on the BBB integrity, as well as mitochondrial potential and reduction of ATP. And there we could also make a nice distinction between the blood side basically and the brain side. Another element of disease modeling that we find super interesting is looking at neuroinflammation, where we add cytokines to such a triculture and then see what the effects are on the barrier again, but also on adhesion of immune cells. So here in the middle panel, you see a monocyte cell line and a stain for ICAM and an adhesion molecule, where we clearly see that by the addition of a cytokine mix, more monocytes start to adhere to the endothelial vessel. And then from there, we also can look at labeled immune cells and where they basically end up in the chip, whether they retain within the endothelial vessel or whether they migrate towards the neuronal compartment. And we can follow those immune cells over time. And what we're looking here in this graph is basically the top channel of endothelial vessels, and then the brain compartment being under the lower dotted line. And you see in the inflamed conditions over time that more and more immune cells start to migrate towards that brain compartment.
Wouter, you could wrap up soon. That would be great.
Yeah, perfect. That brings me indeed to the two last slides. So as a summary, we focused on 3 different applications, and we actually do that in 2 ways. We do that in our labs by providing services, custom model and assay development, as well as compound profiling and screening. And we actually also provide those plates with HBMAC tubules already in it as an organo-ready product, as we call it. We can ship that to your labs so that you can do the work yourself. And this is a testimonial of one of our customers who's using that for AAV transport studies. If you're interested in learning more, check out one or all of those publications or ask any questions in the chat. Happy to answer. Thanks.
Thank you so much, Wouter. That was really interesting. Next up, we are excited to introduce Peter Worthington from Visikol. And Peter, I will let you introduce yourself. If you could share your screen just to make sure we've got it. That looks great. All right, I'll let you take it away. Peter, you are muted.
Sorry about that. Thank you so much. My name is Peter Worthington. I'm the manager of the cell-based assay team at Visikol. Visikol is a contract research organization that focuses on cell-based assays and tissue imaging. Imaging is a big focus of the company. Yeah, so excited to talk to you guys about our blood-brain barrier model. It is a transwell system, 96-well transwell triculture. The 3 cell types are iPSC derived, so we have endothelial cells seated across the top of the barrier, as well as astrocytes and pericytes seated along the bottom. The way that we typically work with this model is we add our test article to the top compartment or the apical compartment or the blood compartment, and then measure the concentration from both top and bottom compartments over time. This allows us to calculate the kinetics of the test article, and from that we can determine the apparent permeability of the molecule. Typically, we quantify the concentration using our LC-MS, but we've used ELISAs, we've used nano-LC as well. Some data around the endothelial cells, and this is from, so the cells are from Fujifilm, and this is imaging data from Fujifilm, if you're interested in making your own model. We've got expression of the breast cancer resistance protein, Claudin-5, the GLUT1 transporter, the multi-drug resistance protein, the transferrin receptor, PGP, the major efflux transporter, zonacecludin, CD98, and E-cadherin. Some functional evidence of the barrier integrity. The first is the TIR, as it's already been spoken about. It is the resistance across the barrier. A higher tier indicates a tighter barrier, and you can see that it increases for the first 5 days, and that's typically when we start our assays. Another example of the barrier performance is a fluorescently conjugated dextran molecule. Without any cells in the model, you can see that it is easily able to pass across the barrier. And if we repeat that experiment with the cells included, that transmission is inhibited. Some further control data, this time with small molecules. So on the left here, we're reporting it as a ratio of the concentration, basically the area under the curve from the LCMS-MS. For the bottom over top compartments, caffeine is our positive control, and we would expect it to pass. So in the absence of cells, it is able to easily pass across the barrier, compared to if we repeat that experiment with cells included, it is still able to easily pass. In comparison to atenolol, so this is our negative control, and we would not expect it to pass. Without any cells present, it is able to easily pass across the barrier, compared to if we repeat the experiment with cells included, and that transmission has been inhibited. Most of our data so far is around small molecules, although we have started to explore some work with large molecules. So just to show sort of functionally that we do get some good some data with large molecules, we've got a transferrin protein that's been fluorescently conjugated, and if we add it to our top compartment without any cells, it is able to easily pass across the barrier. The middle data set here is transferrin with cells included this time, and you can see some of the transmission has been inhibited, but it is still able to pass. And then the third experiment is this time including ferrostatin 2, which is a small molecule inhibitor of the transferrin receptor. And you can see we've again inhibited the transmission of the transferrin molecule. So, so far, the data that I've been showing has been looking at the apparent permeability from apical to basal. What this means is we're adding our test article to the top compartment and then measuring the kinetics as it moves down. We found that this did a decent job translating for certain classes of compounds, but didn't do a good job for all of the compounds that we were testing. So we looked for ways to improve the translatability of our results, and what we came upon was the KPUU value. So instead of just the apparent permeability from top to bottom, we also report it as a ratio with the apparent permeability from basal to apical, as well as correcting the concentration with the free fraction of the test article in the presence of plasma protein or the free fraction in the presence of brain tissue homogenate. And this is the representation of that mathematical model. So we have our test article, and it's either able to be free in the bloodstream or bound to the plasma protein. The free drug is able to pass across the barrier or be effluxed across with the transporter. Once it's in the brain tissue, it's able to either be free or be bound to the brain tissue homogenate. And then the free drug is able to be fluxed back across the barrier. So to determine the bound fraction, we use what's called a rapid equilibrium plate. And what we're basically doing is incubating the test article in the presence of either the plasma protein or the brain tissue homogenate. And using the mass spec, we can determine how much of the test article is free or available. And then if we combine the bound fractions, or the free fractions with our two apparent permeabilities, we have our calculated in vitro KPUU, which is along the y-axis here. And we compared that to some data that we took from literature. So this is our in vivo KPUU. And some caveats for this data is this is from the cerebral spinal fluid. So I was not able to get the concentrations or the ratio with the actual concentration in the brain. This is the closest that I was able to find. But you can see we get a nice regression with an R-squared of 0.77 for this panel of test compounds that we tested against. Yeah, so thanks very much.
Thank you so much, Peter. That was, yeah, really, really interesting. Next up, we've got Deborah Ramsey from SynVivo. So Deborah, if you could jump on screen and share. And Peter, I will take you off. Deborah, you're looking good and I can hear you. So go for it.
Can you see my screen?
Yep.
Great. Thank you so much. And thank you to the organizers for having us here. We're really excited to talk more about our blood-brain barrier model and our organ-on-a-chip platform. Sorry, my slides are not advancing like I'd like them to.
Yeah, I don't see your screen either, Deborah. Maybe try to reshare.
Yes, let me stop sharing and let me reshare. Sorry about that.
No worries. Okay. Yeah, there we go. Looks awesome.
Can everyone see that? Okay, great. And okay, let me see if my slides will advance. Yes, is that good? Okay.
Yeah.
Sorry about that.
Yep, looking good.
So great. So I am the Director of Scientific Development at SynVivo, and we offer a 3D tissue organ-on-a-chip platform where we create in vivo vascularized tissue microenvironments with a scale and morphology and fluidics and cellular interactions. And we utilize these models to better predict the human relevant responses, especially in drug development. And we have a very versatile platform, so we can utilize different libraries of different compound microfluidic chip designs to generate different types of models. And so you'll see on the left-hand side our traditional BBB model, where we have astrocytes and pericytes in the center chamber, surrounded by microvascular endothelial cells that are brain-derived. We can also mimic brain tumor barriers in our center model there with a microvascular bed. And then we can also have a linearized model that allows for efficient drug transport. And we can see here that we develop our endothelial cells using both primary cells as well as cell lines. And you can see on the left-hand side where we've used HUVECs or rat brain endothelial cells. And on the right-hand side, you can see where we used an HC-MECD3 cell line. But this allows us to form a luminized platform so we can dose and deliver drugs through the vasculature, and that you can see the lumen of the HUVEC as it rotates. But our goal for BBB models is to really create a multifunctional neurovascular unit so that we have brain endothelial cells as well as astrocytes and pericytes included together so that we can visualize neuronal processes, utilizing a basement membrane to form established tight junctions, and have the crosstalk that's available as well as a really tight barrier. And so by utilizing our microfluidic platforms, we can create a side-by-side architecture that enables real-time visualization of these cell-to-cell interactions. And then we've got microenvironmental dimensions with an engineered porous gap that allows for immune cell infiltration. I'll be showing some data from that today, as well as enabling us to measure resistance or impedance across these barriers using electrodes and TEER. And so our models, we've done a lot of comparisons with our cell types to try to look and visualize just the tightness of these barriers as well as the cell-to-cell interactions. So you can see on the left-hand side where we've done permeability assays with human microvascular brain endothelial cells, and we can use traceable molecules such as 4-kilodalton FITC dextrans to visualize the tightness of the barrier when those brain microvascular cells are present. And that tight junction protein interaction really increases in the presence of astrocyte-conditioned media or in the presence of astrocytes themselves. So you can see kind of in the middle graph where we utilized both and showed that the tightness of the barriers increased significantly in the presence of the astrocyte-conditioned media. When we visualize those astrocyte endothelial cell interactions, we get the astrocyte infeet forming and touching base with those endothelial cells as shown above. And then again, our models are closely mimicking the in vivo systems in terms of permeability and barrier tightness. We can also look and see how the impacts of having those astrocytes as a co-culture in terms of permeability works as well. So we can see over time that our cells and our human brain endothelial cells are very tight when screened with our 4 kilodalton FITC dextran. But even in the presence of astrocyte conditioned media, we noticed that the co-culture with astrocytes themselves actually create the tightest barrier, as shown in the upper right. And we can also look at the TIR values of that and see an increase in TIR over time. And then we can dose with an inflammatory compound and then change the tightness of that barrier, but then watch the barrier recover when the pro-inflammatory signaling is removed. So that just allows us to monitor these changes in real time and look at the permeability and tightness of the BBB in response to compounds. One of the things that this model has allowed us to do is establish a platform for looking at different compounds that can either influence or reverse the tightness of the barrier once they've have undergone any kind of inflammatory mediated damage. So you can see on the left-hand side, when we dose our human brain microvascular endothelial cells with TNF-alpha, we get a significant increase in permeability. But when we look at molecules that interfere with PKC delta, which is a key element in terms of the pathway of generating a leaky barrier, that interference actually decreases the permeability and restores the tightness of the barrier. And on the right-hand side, we see some compounds from one of our clients and customers that also influences the same type of effect, so that we see less damage in terms of the inflammatory response, and we also see retightening of that blood-brain barrier. And so the platform enables us to screen these compounds and visualize that. In one of our ongoing collaborations with the National Research Council of Canada, we've been looking to kind of optimize the different cell types that we utilize in our SynVivo platform. And they have an iPSC-derived brain endothelial cells, or IBEX, that they have utilized and optimized within Transwell systems, and that data is shown here, has been published. But they've been able to show Claudine and ZO1 expression within their Transwell models. And we wanted to take these IBEX cells and utilize them in our SynVivo platform to look at barrier tightness and for drug screening. One of the ways that the NRC has utilized readouts with really small amounts of material has been capillary electrophoresis-based Western blot, which is known as WES or JES. But for antibody transcytosis detection and experiments, these readouts have been really important in terms of detecting very small volume effluents, and it's also a little bit less expensive than mass spec. We've been utilizing these in our SynVivo platforms to detect the amount of antibodies that have transcytosed across our blood-brain barriers, and this particular readout has been very sensitive and allowed us to detect those methods using microfluidics. And so we assessed 2 different types of antibodies for their crossing of the BBB permeability using these IBEC monocultures in our devices. One of the antibodies was very specific for the BBB, targeting the TMEM38 targets, and the second antibody was a microbiological target, so it's not a mammalian target, and that was served as our control. And so one of the options that we looked at was looking at the permeability of those IBEC monolayers, both pre-transport, so before we added the antibodies, and then post-transport after the antibodies. And these were stained using sodium fluorescein, which is a very small molecule, and showed the tightness of these IBEC monocultures within our SynVivo devices. And one of the interesting aspects about the antibody transcytosis that was observed was that the antibodies that were delivered under flow actually showed an increased permeation across the BBB compared to those that were delivered statically. And then our control antibodies were pretty similar, whether they were delivered static or under flow. So it allowed us to show that our microfluidic models were successful at predicting this receptor-mediated transcytosis. We then developed again, in collaboration with the NRC, we utilized a triculture system. So we had astrocytes and pericytes, as well as the IBEC endothelial cells to form that neurovascular unit, and then examined looking for the antibody transcytosis across those barriers. And we observed, again, both a pre-transport, post-transport looking at the tightness of the IBEC cells, and found them both to be tight when we looked at before and after antibody transport. But one of the interesting things that we saw again was that the concentration of antibodies that were able to successfully cross the BBB triculture increased when the antibodies were delivered under flow. So it allowed us to say both in IBEC monocultures as well as in a triculture that we could visualize that receptor-mediated transcytosis. In collaboration with the NRC, we also did some studies with CAR T cells, which allowed us to look at the extravasation of the CAR cells in response to a glioblastoma signal. This platform was really great in terms of looking at immune cell extravasation into the glioblastomas, as well as real-time quantification of the killing effect on those cells. And you can see the CAR Ts as they're being delivered through the IBEC monolase, and you can see the stained glioblastoma cells above and below. And so one of the things that we observed with the CAR Ts is that they were successfully able to adhere and arrest onto the IBEC cells in response to the signaling from those glioblastoma cells. So we were able to successfully observe the leukocyte adhesion cascade using this cell model. And then we were able to also visualize the extravasation of those CAR T cells directly into the glioblastoma channels. So you can look at the circle that's where extravasation is labeled, and the yellow arrow indicating down, the cells are permeating through and sliding down to glioblastoma channel. And we were able then also to evaluate the permeability of the IBEC monocle IBEC layer within this glioblastoma IBEC co-culture system. And before CAR T cell delivery, we saw that the IBEC monolayer was very tight, but afterwards the fluorescence intensity increased, indicating that the CAR T treatment was generating some permeation and permeability. And that has been observed clinically as well. So we were very excited to notice that our SynVivo model was able to assess not only the blood-brain barrier integrity before, but after immunotherapy treatment. And so once again, in terms of visualization, that blood tumor barrier model allowed us to also observe what was happening to the glioblastoma cells following CAR T administration. And you can see on the left-hand side that the red signal from the red fluorescent proteins expressed by the glioblastoma drastically decreases within 48 hours. And if you look at the phase overlay on the right-hand side, you can see that the cells are apoptosed and have been killed by our CAR T cells. So we were able to then visualize the immune-mediated tumor cytotoxicity. So in conclusion, we have two SynVivo models that we offer. They include the blood-brain barrier, which is that neurovascular unit that shows tightness and can look at drug screening for different effects on the BBB. But we also offer a blood tumor barrier, as shown with our CAR T work with NRC and in service to glioblastoma. We also offer different services that assess BBB permeability, screening compounds, and the delivery of those immunotherapies across the barrier. So I'd like to acknowledge our collaborators at the NRC Canada, especially Dr. Anna Jasiewski, and we're located in Huntsville, Alabama, and I look forward to any questions you may have. Thank you so much.
Thank you, Deborah. That was, incredibly interesting and, really useful. Next up will be Dr. Michael Shuler from Hesperos. Dr. Shuler, can you share your screen?
I'm trying. Let's see. Does this work?
Not yet.
Okay, I'm hitting share. Let me hit this thing again. Not now. Shoot. It's disappeared.
So just to the left of the leave button on Teams should be a share screen button.
Yeah, I hit the share screen button. And that actually is what messed things up. Let's see.
Okay, if needed, I can share your slides and you can just let me know when to advance. Oh, I think you got it.
Okay. And this is a little different than, this is not the talk though, this is the problem. I had it all set up very nicely, and now it's not working.
That's just how technology works.
Okay, well, the talk you have is not quite as good as the one I would have given, but let's go with it.
Okay.
Let's see, is there anything happening?
No, nothing has changed on your shared screen.
This is talk I have, but you can't see it, right?
I can see it.
You can?
Yep.
Okay. This is the one I wanted to give. Okay.
We see Hesperos BBB slides. Is that what you're trying to share?
It says the first slide says microscale microscale human BBB models and multi-organ models. Did you see that?
No, we don't. I'd recommend you stop sharing and try again.
Okay, let's cancel that. And I hit the share to stop sharing. It keeps bringing up another thing for me. So it's
so there should be a little window with an X.
Sorry.
Yeah.
You did send me your slides, so if you need me to pull them up, I can do that.
Okay, probably what we're going to have to do because I can't see anything here. I can see my slides, but you can't see them.
Okay, I stopped sharing for you, so you should be able to try one more time. And if that doesn't work, we'll go ahead and have Matt share for you.
See, it's probably not going to.
If it doesn't work, we'll blame the weather in Ithaca.
No, that one's not it either.
There you go. I can see the slides that I've got.
Yeah,
that's looking good.
That's not the wrong talk. The one that I had didn't come up, so that's not the right talk. And I had it over here on my screen to bring on up, and then it. Let me try one other thing, and it's just disappeared basically. So why don't you put up what you have? I stopped sharing.
Yep, this is the slide set that you sent me, so should be good to go. Just let me know when you need me to move on to the next one.
Okay. Okay, so sorry about that. I'm going to talk since Jay Hickman, who's really the person involved, was supposed to give it, he's unable today. I've been associated with Hesperos since its founding. I'm also a professor emeritus from Cornell University. Next slide. So the overview of Hesperos, the important thing here is that we're talking about multi-organ systems based on the PBPK. So we worry about body and a chip. We use the serum-free medium in everything the company does. We have a pumpless system. It's self-contained, no external pumps on a rocker platform back and forth. We worry a lot about functional readouts. TER is a functional readout. But we also have readouts, microelectrode arrays and fourth generation. I'll show that in just a second. Our company's growing now to about 45. We're a CRO. And I think probably one thing that's of interest is the first microphysiological system data that's been used in an IND application by Sanofi has led to authorization of phase 2 clinical trial for a rare disease. So this system has actually advanced to the point where at least in one case has been accepted for use in a clinical trial. Next slide. Next. We've built a model of a number of different barrier tissues, blood-brain barrier being one of them. Let's just go to the next one. this is some work I did at Cornell, and it was actually transferred ultimately to the company. We built an IPS induced pluripotent stem cell derived brain microvascular endothelial. Sean Palachuk, Eric Schuster at Wisconsin did that. Co-culture with astrocytes, porous membrane. Next slide. And we have an unlocking platform. Again, there's no inlet and outlet. There's 2 reservoirs here. And the brain microendothelial cell layer is indented from the flow. That's to prevent get the right amount of shear in the cells. Too much shear is damaging. Too little shear doesn't work so well. The residence time of blood in the brain is 2.3 minutes. This mimics that as well. Next slide. We can obtain, this is back in 2016, published in 2017. We can maintain for a fair amount of period of time the authentic tear in the brain with a combination of astrocytes and co-culture. And this just shows we get the tight junctions. Next slide. This shows that there's a pretty good correlation between permeability of the BBB that's measured on chip versus what's known in vivo for a range of 3 compounds with quite very significantly permeability, plus physiolabeled dextran. So we're able to get a pretty good correlation that way. Next slide. Now, we want to put this in the multi-organ system. It's possible to look at the drug effects on BBB and side effects on other organs. It's one of the things we want to do. We'll look at evaluation of potential routes of administration. That's mostly what I'll talk about today. We'll also look at interaction and combinations of drugs, another thing we do. Next slide. The Cornell BBB model I showed you, it doesn't work directly with the Hesperos system. It has to work in a serum-free media. We have 1% serum in the Cornell model. And also we used rat astrocytes. Now we're using human astrocytes that are available from this company right here. And we really asked, can we combine the BB with the CNS component? Next slide. We do have these clinical relevant readouts. We can measure muscle contraction. We measure electrical activity. We measure the amount of things developed by the liver. But they're all functional, and they can be done very quickly, so we can get responses in terms of minutes as opposed to hours and days on many of these systems, particularly in terms of neurological effects and contractibility of muscles. Next slide. So this is the system we're working with: IV administration, oral administration, intrathecal administration, blood-brain barriers over the CNS system. Next slide. And basically, this just shows that the culture, we're just using IPS and astrocytes here. We've talked about doing parasites, but this works reasonably well for us, and kind of modify cost versus authenticity. This shows we get tight junctions in the BMC, we get expression of the appropriate proteins in the astrocytes. Next slide. And again, this is the GI insert. I didn't have a chance to mention here, but the GI insert, we have actually a fairly complex model. We use a simpler model here with myofibroblasts, KCO2, and HT29 goblet cells. But it still is probably sufficient for what we're doing, although we have a much more sophisticated model we built. And this just shows you get the tight junctions. A little bit sparse here because when they took it out of the system, they didn't do a good job. But anyway, you can have tight junctions all the way through. P-glycoprotein, GluT up here. And next slide. It just shows that the BMCs that we're working with have an appropriate GLUT1 activity. In the absence of an inhibitor, they transport this reporter compound for glucose into NDDPG. If we add the inhibitor, it essentially blocks it, which is what we expect would happen. And this is due not to a change in the permeability of the system, but actually in terms of the GLUT1 transporter. Next slide. And this shows PGP-mediated transport in the BBB. Again, this is got messed up on this slide, but it shows there basically as an efflux promote pump. So you expect higher levels of the system when the efflux pump is inhibited. So if we put verapamil in there, which inhibits the pump, we have a higher exposure of the compound bicuculline at 10 microliters in between. No PGP inhibitor, then this is what we see, and the efflux pump is working well. And the tight junction persists both pre and pro. Next slide. Next slide. So, okay. This is just shows that we can operate some in the CNS side. This is cortical neurons. In those cases, we can sustain a reasonable level of the BBTR and the tight junctions in the GI tract. GI tract kind of matures with time. During 7 days, we have a little loss of the BBB activity, but it's still not too bad. Next slide. This shows that the drugs don't affect the pre-dose and post-dose, really don't affect the TER values for both the BBB and the GI tract, and this is done for caffeine, bicuculline, and the canic acid. Next slide. This shows, again, caffeine goes through very quickly, so the difference between oral and IV is not very much. It goes through a lot less quickly for bicuculline and canic acid oral dose. And as a consequence, we see a difference in the oral and IV doses depending on the particular compound we have. Next slide. So we do have a BB expresses tight junction proteins, and it's functional with a physiological meaningful multi-organ model. And that's probably one difference between what we've seen in the prior talks and this one is that we do work with a multi-organ system. We can put in cardiac system, we can put in liver, we can put in bone marrow, we can put in all sorts of other tissues with this if we wish. We maintain transporter function at least for 7 days. The transport profile depends on the compound as we expect. And the barrier integrity is maintained pre and post drug. And I'm sorry that the real talk didn't get to be up there, but this is, I think, hopefully it'll give you at least an idea. So thank you for your attention.
Thank you, Dr. Shuler. Yeah, my apologies for any technical difficulties, but it was very interesting to hear the difference in compound transportation. So I will stop sharing my screen, and last, but certainly not least, should be Leopold from Tissuse. Leopold, can you share your screen?
Yes, let's try it again. Because it wasn't working earlier, but let's give it another try.
I can see it.
You can see it?
Yep.
Okay, that's great. So it seems to work this time. Amazing.
Yeah, can I get one more person to confirm?
Yeah, it looks great.
Okay, thank you.
So thank you, first of all, for the chance to give a presentation here on what we are doing in the field of the blood-brain barrier. I think my talk fits quite well with the talk we just heard from Hesperos, because also we are working on multi-organ combinations. So I'm talking here today on the behalf of Tissuse. So we are a Berlin-based MPS developer which focus mostly on multi-organ chip combinations. And the blood-brain barrier we have been modeling in our so-called Humimic Chip 4, which is a cultivation platform, as the name basically says, for 4 organ models in parallel. So our main idea here was to build basically a system that also works with an autologous donor background. So in the past, we have been working with various models. Our system is in general quite flexible. We can put in different commercially available models or also self-developed models and combine them in different manners. But for the future, we are more and more progressing and trying to build systems that are really derived from one common donor. And we use iPSC technology to differentiate iPSCs into different organ models and then combine those in our chip systems. And what we used for the BBB was a rather simple setup. So we started with a transwell model, as you see here in the bottom. So we used the standard 96-well transwell, and we combined brain microvascular endothelial cells derived from iPS, same protocol as we saw before from Schuster and Lippmann, and we combined it with neural spheroids that we cultured inside of the transwell. And back in 2019, we published for the first time this Humimic Chip 4 with a combination of an intestinal barrier, a liver spheroid, a kidney barrier, and a neural spheroid model. And having that as a basis, we then progressed in also including a BBB model in that chip system. And what we published last year was a combination of a BBB brain model with liver spheroids. I will show you some of that data today. And also we have progressed again and basically assembled a whole chip 4 with all 4 models, which now also includes a BBB brain model. As I said, the basis of all cell models that you see in that presentation are our own set of iPSC. So we have a bank of 6 donors currently, which where we have the full consent for commercial use, so we are free to use them. They are quite broadly characterized, so we have flow cytometry, qPCR data, SNP genotyping, RNA-seq data, pluritest, and so on. So they are characterized by the state-of-the-art, I would say. And this is basically the basis for the different protocols that we apply to differentiate them. For the neuronal part or the neural part in our BBB model that sits inside of the transwell, we use a bioreactor differentiation protocol, which differentiates iPS in spheroids directly into so-called neural spheroids. You see here, after 21 days, they show these quite nice neural tube-like structures. But after 3 weeks, they are mostly early progenitors in neural stem cell, of course. But we see that they have some progenitors already in the glia and neuronal compartment. Those can be further matured for quite a long time. So we have matured them up to 200 days. Until 120 days, we still see progressing maturation of them. We see differentiation of astroglia also after some time, and when we combine them with the blood-brain barrier, we always use them at time points where they already have astroglia, so that the astrocytes basically are present in the model. And we know that they differentiate both into glutamatergic as well as GABAergic neurons. So for the BBB, this basically is some basic characterization of these brain microvascular endothelial cells that we use. We have seen similar images already from the previous speakers. So this is like state-of-the-art differentiation protocol from iPSCs. So the nice thing about them is they form these really tight barriers, right? You have really high tier. We see really low sodium fluorescein permeation, below 1 micrometer per minute. They have minimal albumin permeation properties. And they express also various relevant tight junctions such as Claudin 5 and occludin, and also transporters such as GLUT1, MDR1, and BCRP. What was also said before and what needs to be kept in mind for these cells is that they have some epithelial fingerprint, so they are, or they should not be called pure endothelial cells, which is why they're also called nowadays endothelial-like cells. So what we see is that they also express levels of E-cadherin alongside with the VE-cadherin, opposing normal endothelial cells differentiated also from iPS. But on the other hand, they have much stronger barrier properties than these classical endothelial cells, so they have this really high tier and this really low permeation for most compounds. We combined that then with a liver-derived model. So on the left side, you basically see the BBB neural model composed of these spheroids as well as these monolayer brain microvascular cells, which are on the bottom side of the transfer. And we combined them with an iPSC-derived liver model, which is spheroid-based and which consists of hepatocyte-like cells, stromal cells, and endothelial cells in a near physiological mixture. And those are combined in our so-called human chip 4. So what you see here is only the fluidic layer. You see these compartments here, which are the intestinal compartment, the brain compartment, and the liver compartment, which are interconnected by an on-chip micropump and a circulation system that basically recirculates media inside of that system. For that brain-liver co-culture assay, we went for a co-cultivation time of 7 days, as those was the time that we found the BBB cells rather stable on the chip. Afterwards, the tier and the arrangement more and more declined. But for 7 days, we found it to be quite fine. We left them in there for a 5-day adjustment phase first, and then used the last 2 days of the assay for treatment with compounds, as we'll show in the next slide. We also saw that the barrier basically was maintained here in our chip system. We see that for small molecules such as glucose, there's basically no difference whether these brain microvascular cells are there or not. So glucose still reaches the brain compartment. But for larger molecules such as albumin, for example, we see that pronounced drop basically as soon as these cells are contained. We don't find so much albumin anymore in the brain compartment as opposed to when these cells are not present. And the exciting thing about combining organ models is that you can then also study basically the interaction, or you can basically model or investigate secondary effects. So what we tried to do, or what we did, was to apply 2 model compounds, atenolol and propranolol, which are 2 beta blockers, but which have different properties of passing this BBB. So while propranolol is a molecule that quite easily passes the BBB, atenolol rather does not. And this, first of all, we saw through the addition of the BMECs. So what we see here without BMECs, you see this is this ratio basically between the circulation and the brain compartment. And if there are no BMECs, then basically both propranolol as well as atenolol are close to 1. So the concentration is similar between both compartments. As soon as we add the BMECs, you see that basically atenolol is not passing so much into the brain compartment with a brain plasma ratio which is close to in vivo data, while propranolol here it's still quite easily passing into the brain compartment. In vivo, we see that the ratio is even much higher, so the compound really accumulates in the brain in vivo. But we have to keep in mind that this is only this 2-organ combination, and this setup basically has no chance to excrete the compound, which would happen in vivo. So this only is now the BBB part of the story, but where it gets interesting is when we look also on the combination with the liver model, because also the liver model is active and metabolizes both the atenolol and propranolol. And when we looked on different metabolites, we found, for example, the glucuronidized versions of the atenolol and the propranolol. And these have then different properties as compared to the parent compounds. So when we first look on the propranolol, and keep in mind that the parent compound could quite easily pass into the brain compartment, we now see that the propranolol glucuronidide, when the BMECs are present, is barely found in the brain, so the metabolite is blocked from passage into the brain compartment. And interestingly, for the atenolol, we find it quite highly in the brain compartment when the BMECs are there, which we believe is due to metabolization in the brain, as it is known that also glia cells can metabolize atenolol. And we think that basically the reaction is happening here in the brain compartment, and that the metabolite is then trapped basically behind the blood-brain barrier. As I said, we also recombined the 4-organ setup of that chip basically with a BBB brain model, an intestinal model, a liver spheroid model, and a kidney model. The basic setup with that 7-day culture phase was kept similar to the previous assay, so 5 days adjustment, 2 days treatment, this time with a daily media exchange due to the higher need of medium for the 4-organ models. And we used application of atenolol, carbamazepine, and verapamil this time as model compounds. First of all, we could see that both the kidney as well as the brain barrier were rather tight. So we saw that albumin ratios compared to the albumin concentrations in the overall circulation and also in the intestinal compartment was quite low, both in brain compartment as well as in the kidney circuit. And we saw that also, since we had really high glucose consumption by the brain model, glucose ratios was lower than in the rest of the circulation. Then this time, we also looked on the recovery of the compounds. So we found that verapamil was rather challenging in our chip system. So the recovery means basically how much of the compound is lost into the PDMS. And the verapamil, we saw that, as expected, it actually had a quite high binding. So we also only got out a rather small fraction of the verapamil, which was challenging. We could also look at the intestinal absorption. So basically how or which extent of the compound passed through the intestine. And when we compared that to in vivo data, we saw that probably the intestinal barrier is not as tight since it is iPSC derived as an in vivo situation, but we still see the same ratios, with atenolol having the lowest permeation and verapamil having the highest permeation through the intestine. When we looked at the brain plasma ratios, so basically how well the compounds distribute between the circulation and the brain compartment, we again see that atenolol has a rather low value of around 0.2, which is close to in vivo. Carbamazepine is close to 1, also similar to in vivo. Verapamil is actually higher, so in vivo it is described to be rather at 0.5. Here we find rather values close to 1, which could either mean that the PGP activity is not as high as needed, but it could also be due to the rather high binding of the verapamil in the circulation that this value is skewed here. Beneath, so in addition to brain and circulation ratios, we can also calculate the PAP values. We calculated this for the earliest time point that we measured. And also here, PAP values are, we compared with a similar in vitro assay, and we also find similar correlation. So with atenolol having the lowest permeation ratios and carbamazepine having by far the highest permeation ratios. So a short outlook where we want to go next. So what is definitely on the table currently is that we have released last year our so-called Human Act Sense, which is a platform for electrophysiological stimulation and readouts. So that basically enables tear impedance, things like ECG and stimulation of tissues. And so far we are using it with commercially available electrodes. So it's more or less a tear and impedance device, but we are currently working on also integrating on-chip electrodes so that in future we are also able to directly measure things like tear, or to have electrical stimulation directly in our chip system without manipulation of the models. And some broader perspective and outlook, which from our perspective are interesting fields to further develop these kind of combinations, or where this could go. So in terms of increasing the complexity of the BBB brain model, definitely addition of parasites and microglia is still a big topic. We think that biological membranes will make a big difference in regard or in comparison to the PET or PC membranes that we currently use. And I would also like to look into alternative differentiation protocols from iPSCs to omit that epithelial fingerprint that we see in the current differentiation protocols. Then, of course, disease models are super interesting. So this can be modeled from iPSCs, this was shown before, to model, for example, degenerative diseases such as Alzheimer's or Parkinson's. And as a more general scope of our company, we are also increasing more and more the complexity of our chip system, and we really want to go further and further in the direction of a human on a chip concept. So with that, I would like to close my talk. Thank you so much for listening in here today. And I guess we are progressing now to the Q&A session, right?
Yes, thank you so much, Leopold. We will progress to our question and answer session. I will bring all of our speakers on screen. And we've got a number of questions in the Q&A. The first of which is, I believe, for Dr. Ramsey. No, I apologize. This one looks more general. So what is the level of technical skill that is required for your target customers? What barriers do you experience when trying to pitch this to researchers, especially those who are unfamiliar with microfluidics? And it looks like Dr. Hughes has raised his hand. You should all be able to unmute yourselves as presenters. So,
yeah, the biology is the hard part of this. The widget part, making the platforms, is fairly well understood at this point. So we do all stuff in house, and I know many others do too. So there really is no barrier to entry for a customer. As long as they're willing to send their compounds, we can do the studies. At some point, we may send out a platform to customers, but we're not currently doing that. So it makes it an easy entry for a customer by operating as a CRO.
Okay, thank you. Leopold, do you have any input on that question? The technical skill required to enter into this field?
Can you specify a bit, like, is it more like towards a completely new user of MPSCs, or?
It is as specific as the question was asked.
Yeah.
So what barriers to
I guess
what barriers do your clients experience when entering into microfluidics?
So I would say for our systems, the progression is relatively easy. I would say it depends a bit on, of course, where you are starting. But we set up, for example, our chip system that it is relatively easy to incorporate models that you're already using in the lab. So if you're already working with like spheroid models, or if you're working with transfer models, you usually, for most of these models, it is quite easy to just incorporate them into our system and then to culture them in there. Overall, of course, I mean, there's some knowledge that need to be learned, but this usually can be trained rather quickly in the training, and then it is rather, I would not say easy, but it is definitely possible for every biological lab to adapt this technology at this point of time.
Thank you, Leopold. Wouter, do you have any input on the question?
Yeah, indeed, I think it's a good question, and it definitely requires a bit of training to get used to working with microfluidics in general, I think. And I think there are a lot of solutions there. So for some, it's indeed very helpful to just ship the compounds to the service provider, basically, any of these companies. And for others, it's also possible to work with the system yourself. And we, for the BBB work, we also generated plates with cells that we can ship. So then you don't have the issue of doing biology yourself. You just need to add your compounds and do the sampling. So that takes away one of the hurdles. And I think it could be a very interesting way of working with these kind of systems.
Thank you. Dr. Ramsey, any input on that question?
Yes, I think it's a fabulous question. When I was trained in cell culture, and I didn't come to microfluidics until kind of later in my career, and I found that one of the things that helped me was to just think a lot smaller than what I had thought of before when I was using my well plates. So, you're kind of working into a much smaller space, but then you can also observe how your cells then respond physiologically to that. And we, as a small CRO, we offer a lot of technical support and protocols like the other, colleagues had mentioned as well. And that helps new users become, more willing and more able to tackle, if they're new to microfluidics and kind of tackle these new techniques, and then it just becomes something that they integrate into their regular tissue culture practice.
Yeah.
So it becomes something that you grow very familiar with, just like all tissue culture. So I encourage everyone to try, because it's definitely gives you the readouts that are really important for your research. It moves your research forward.
Great. Thank you, Deborah. Peter, any thoughts further than the rest of the group?
So we don't work with microfluidics, but I would say a main barrier is how do you interpret the results, and what does the FDA think about the results? And I don't always have a good answer to that question, but I would say that's the main barrier that we experience.
And that is one thing that the North American 3 Hours Collaborative is trying to get consensus on, is what the FDA thinks about things. So yeah, we are happy to hear that feedback. And then Dr. Shuler, last but not least.
Yeah, so Hesperos has always operated as a CRO. The feeling was that the combination technology is such that we're not sure that all our customers will get reliable results otherwise. A lot of it is also based upon the fact that a lot of the functional readouts we do are not necessarily that standard. And doing it in the context of these systems and making sure that you have good control over the solutions, the absorption, the surface chemistry, everything else that goes into it, so that the results you get are meaningful, actually requires a fair amount of experience. So we prefer to do it as a CRO.
Okay, thank you. We've got a few company-specific questions, which I will forward to those companies. But the more general ones, let's see. Well, I guess they're all a little bit company-specific, but I will try to generalize them.
Matt, you can make them all company specific. It's totally fine for,
okay.
Yeah. And then if other people want to chime in, it's all good.
That's my boss letting me know how to run this meeting. So I guess then question for SynVivo. Can this system be used both in the dynamic flow with pumps and static flow? And are the cells seated manually? And then they said, thank you. Khan said, or Munzarine Khan said, thank you at the end. So a very polite question.
Oh, thank you. That's a great question. So to answer, I'll start maybe from the bottom and make sure I answer the other parts. So yes, you can seed our microfluidic chips manually. You can also use syringe pumps if you want to, but you can seed them manually. So that hopefully that will be good answer to the question. And there are other when he's asking about can these systems be used statically or under flow, and the answer to that also is yes. But I say that with a caveat, because the barrier tightness in our blood-brain barrier model is really enhanced under flow conditions. So I think it just depends on the questions that are needing to be answered when you're setting up your experiments, but these systems can be used statically or they can be used under physiological fluid flow. Thank you for that question.
Does anyone else have any thoughts on dynamic versus static flow and company capabilities?
Just dynamic flow is, I think, what the cells typically see. So if you match what's their physiologic, that's going to be more accurate response.
Yeah, I agree with Mike. I mean, we see quite dramatic changes in gene expression with cells under flow. There's critical receptors that aren't even expressed in static culture that come up under shear conditions. So I think having flow over the endothelial cells makes sense.
Thank you, Chris, and thank you, Mike. Wouter, you've already responded to this one, but we want to get it on video. Have you measured neonatal FC receptor expression level and stability over time in the Mimetas BBB model?
Yes, I will give the same answer as in the chat. So we did look at that, but not too extensively. So we've seen it, but more in a black and white matter. So we could say yes, it's there, but we haven't looked at it over time and looked at stability yet. Could be really interesting to look at as well. Oh.
And another Mimetas question that you have already also answered in the chat, but on video is better. To correlate TEER with viability, I wonder if your platform has an option to measure viability at different time points in the same plate by the media collection or impedance or other methods.
Yes, indeed, that we can nicely multiplex at different readouts. So, for example, looking at tier values as well as on viability assessment. And viability assessment can either be done with taking samples, looking at LDH release or WST8, or looking at imaging-based readouts. And there, what we typically see is that the tear, the barrier disruption, is something that is seen a bit earlier than actually viability decrease. So sometimes we see that the tight junctions start to loosen, but the cells are not dying yet. So it could indeed be super interesting to combine those readouts and look at the specific effects of your compounds. Some might kill all the cells basically, where others might have a more specific effect on the tight junctions. And indeed by combining those readouts, you can specify that.
Thank you. I will open the question to the rest of the group if there are any more thoughts. Correlating tier with viability. If there are methods that you're using that are worth mentioning. All right, hearing nothing. We've got a couple more questions that are not very specific. Asking about assays with brain organoids, but I'm not sure if the question asker would like to follow up and give me more specifics on that one.
I suspect, Matt, that like everyone, they're interested to know whether you can vascularize brain organoids and whether you can see blood-brain barrier function in organoids. And I suspect that most of us are working on that. I know we are. So yes, we can put brain organoids into our platform. We can put iPSC-derived organoids, see those differentiating. We're currently looking to identify the kind of vasculature that is associating with those organoids, but that's ongoing work.
Thank you. And there is a follow-up question for you. Are the cells in a gel-based layer? If yes, how much does the gel contribute to the permeability? For example, do you have gel-only permeability data?
If that's a question to me, it's not an answerable question. I mean, without vessels, because we generate vessels, we generate a vascular network. We're not lining a channel. So if you have a channel that you're lining, yes, you can ask what the permeability of that is in the absence of endothelial cells, but we're actually generating a capillary network. So it's an unanswerable question as to what the permeability would be in the absence of cells. But that said, we do make an effort to generate a matrix that is appropriate for each of the tissues we use. So the matrix we have in our brain model is tuned to what you find in the brain. How much that contributes to barrier properties, I don't know, because what we find is in our system that the matrix gets rapidly remodeled anyway. So there's a lot of crosstalk between the cells, between the endothelial cells and the parasites. They both contribute to formation of a complex basement membrane. And then the tissue outside of that, again, is going to be remodeled by the astrocytes, the neurons, the parasites. So what you start with is not what you end up with. But what you end up with is presumably what the cells would like to have.
Does anyone else have anything to contribute on that question? If not, I would like to turn it over to David. If you've got anything to add about IQ so that we can advertise that organization before we close out, that would be great.
I don't have any updates, but yeah, I just want to thank all of the speakers. Great. So,
David, you're really quiet for me.
Same.
I also don't seem to have the ability to bring you back on screen, which is weird.
Hold
hello?
on.
Can you hear me better?
Yep.
Okay. I think I forgot to put down the mic. Yeah, I don't have any big updates. But yeah, if anyone's interested in learning more about the IQ MPS, they could check out our website or email me directly, and I'm happy to answer that or point you in the right direction. But yeah, thank you all the speakers for coming today. I think we had some great talks.
Thank you, everyone. That was excellent. Bye-bye.
Thank you.
Well,
Bye.
I will. Oh. Sorry, I will finish up by just advertising our Microphysiological Systems Technology Hub, which has recently been updated. You can see previous presentations, workshops, all on our website. And yeah, I just want to thank everyone for attending. We really appreciate your presence and your contributions. And we will be back in a couple months with our next big webinar. So stay tuned. And with that, I will sign off. Thank you.